The next talk is Vinicius Maracaja Coutinho on single-cell transcription landscape of coding genes and long non-coding RNA orchestrating mouse heart development. Vinicius, your stage.
Hello, everyone. First of all, thank you very much, the organizing committee, for the invitation to be here and the opportunity to show a little bit of what we are doing on single-cell genomics and transcriptomics. This is an ongoing project developed by the PhD student Thais Ramos, who is co-advised by myself and by Dr. Thais Dorrego from the Universidade Federal de Paraíba, and in collaboration with Dr. Sergio Lavandeiro from the University of Chile. According to different sources, the heart and cardiovascular disease, they are the main cause of death globally in both developed and developing countries. Additionally, in a variety of pathophysiological conditions, the postnatal heart returns to the fetal gene program in a way that different genes related to the developmental stage, they are re-expressed in a variety of different cardiovascular diseases. Also, it's known that the majority of the eukaryotic genome is transcribed in a sea of non-coding RNAs, with only a small proportion of the transcripts being translated into proteins. And these non-coding RNAs, they are known to regulate different key biological processes in the cell, and the deregulation of these transcripts can lead to different diseases, including different cardiovascular diseases that can lead to heart failure. In 2018, in a collaboration with the group of Tony Kozarides, who participated in the description of a new type of long non-coding RNAs called the positionally conserved non-coding RNAs, or pcRNAs. The long non-coding RNAs are known to act by recruiting chromatin-modifying enzymes in order to regulate the expression of the neighborhood genes. And in that work, we used public data sets from the ENCODE project in order to identify all the non-coding RNAs available in the both human and mouse genomes that could be potentially positionally conserved or synthetic. They are synthetic between both genomes. For that, we looked for coding genes in human that they possess an orthologous at the mouse genome, and identified if you could have a long non-coding RNAs in the vicinity of this coding gene that possess a promoter region conserved between both transcripts. As we know, long non-coding RNAs, they are not well conserved when we take into account the primary sequence. So using this approach, we're able to identify synthetic non-coding RNAs or positionally conserved non-coding RNAs between both genomes. Here we can see the whole pipeline that we used to identify a set of 1,700 positionally conserved RNAs that are long non-coding RNAs synthetic between both organisms. Also, we found that these non-coding RNAs, they overlap the motifs related to the CTCF complex that are key for the topological regulation of DNA. And these long non-coding RNAs or RNAs, they are enriched in these regions in a way that they could be participating in this topological regulation. Also, we found that these long non-coding RNAs, they are located nearby genes that are related to development, including different cardiovascular tissues, as we can see here, artery morphogenesis and smooth muscle cell differentiation. Also, we found that these non-coding RNAs, they present the expression pattern and a tissue-specific expression pattern with some of these long non-coding RNAs being expressed specifically in heart. Taking into account these antecedents, we decided to explore the single-cell expression pattern of these non-coding RNAs along heart development with the goal of identifying different transcripts that could be markers for different cell types, and also that could be key genes, key long non-coding RNAs associated with genes related to the fetal gene program, and in a way that we could in the future associate these transcripts with different cardiovascular disease as transcripts that could be reactivated post-birth. To perform this analysis, we integrated public data sets related to single-cell heart development, specifically from this manuscript, in which they were able to generate more than 1,300 RNA-seq data from single cells, in which they were able to identify only 3 cell types, and where it's worth to note that they only focus that they work in protein coding genes. To perform our analysis, we first defined the set of different reference transcripts in which we use the gene code for the coding genes. And for the long no-coding RNAs, we integrated 3 different data sets from gene code, ensemble, and the manuscript that I mentioned before. We used all this pipeline in order to estimate the expression of these transcripts, but I will not get into details because of the time. And this pipeline, in order to identify the different cell types and markers associated to each different cell types that we identified along this process. Here you can see the number of cells that we are working on. Here you can see each line, we can have a different time points along the cell development, along the heart development. E.9.5 represents the embryonic stage 9.5 days after 9.5 days. And here the P21 is postnatal. So we can have a 21 days post since birth, after birth. Here we have the number of cells. Each column is a different compartment of the heart: ventricle, atrium, right ventricle, left ventricle, and left atrium, according to the different colors here. And it's worth to note that at the embryonic stage 9.5, the heart is not yet divided between right and left. So we only have the ventricle and the atrium without this division here. And also it's worth to mention that only for left ventricle, we have cells for all the time points along heart development. Here we can see the number of transcripts that we identified for each compartment of the heart without differentiating the cell types that we have here. We're able to identify thousands of long no-coding RNAs and hundreds of positionally conserved no-coding RNAs in the different compartments. And these numbers are in accordance with the numbers of long no-coding RNAs that are published in literature related to long no-coding RNAs analysis using single cell data. Here you can see the cell types classification according to the expression patterns. Each figure is a different time point along development and a different heart compartment. Each color is a different cell type, as we can see here. We were able to identify 8 different cell types, much more than the original publication, which were able to identify 3 cell types. And here we are also able to identify 2 subpopulations of cardiomyocytes, here in the red asterisks, and 2 subpopulations of macrophages at this time point here. We are also able to identify several novel cell marks for the 8 cell types that we identified, including different markers for coding genes, long no-coding RNAs, and pcRNAs. Each line here, we have a different heart compartment and a different time point along development. Each column is a different cell types, and each circle here is the transcript type that we're able to associate as a marker. For example, here in blue, we have the coding genes. In yellow, the long no-coding RNAs, and in red, the pcRNAs that were identified as markers for each different cell type. We're also to identify a specific expression signature for coding genes and long no-coding RNAs for the same cell type at different heart compartments. For example, here, for this time point here, embryonic stage 14.5, we're able to identify 163 long no-coding RNAs that are expressed in cardiomyocytes, but specifically at the right ventricle. Meanwhile, here we have the 46 long no-coding RNAs that were expressed in cardiomyocytes, specifically at the left ventricle. Next, we used co-expression data, co-expression modules analysis in order to associate the long no-coding RNAs with different coding genes that could be participating as key genes of the fetal gene program. For that, we take into account the idea that transcripts that are systematically co-expressed, they tend to participate of the same biological process. As we can see here, the model, the network 19 here, we have in blue coding genes and in red long no-coding RNAs that are participating in protein polyubiquitination. So we used this approach to identify different models of transcripts that are co-expressed for each heart compartment at each time point of development. And here you can see the network for a particular time point where you can see coding genes and long no-coding RNAs that are co-expressed. And here you can see that this model, they are enriched with genes related to vasculature development, angiogenesis, blood vessel morphogenesis, among other biological processes. Here you can see a gene set enrichment analysis of this model, of these transcripts according to different cell types. For example, here you can see the two different cardiomyocytes populations. Here you can see endothelial cells, and here you can see fibroblasts. Here we can see the same analysis for different time points and heart compartments. Each line represents a particular model of interest. Each column is a different cell types, and each figure is a different cell heart compartment and a different time point. Here you can see the networks related to each model, the gene set enrichment analysis, and the overrepresentation of the biological process related to these models. And as I mentioned, this is an ongoing project, and the conclusion so far is that we are able to identify novel cell-specific markers for coding genes, long-non-coding RNAs, and pcRNAs along mouse heart development. We are able to identify that these transcripts, they present a specific expression signature for the same cell type at different heart compartments, and we're able to identify long-non-coding RNAs potentially associated with key genes related to the fetal gene program. As next step, we intend to perform time course analysis using cardiomyocytes from the left ventricle, the unique, the heart compartment and the cell type that we're able to identify along all time points, and to cross-reference the data here obtained with public and in-house generated single cell data from different heart disease in order to associate these long-non-coding RNAs related to the fetal gene program with some key cardiovascular disease. This work was developed thanks to this amazing team, especially Thais Hatz Ramos, and with the funding from different Chilean agencies. Thank you very much.
Thanks a lot, Vinicius. Also, we have Vinicius now in the live chat. Maybe I can kick off with one burning question that I had. Because working with long non-coding RNAs, we have, or you will have the challenge of, you will face many challenges in terms of annotation, but also working on single cell level. Many of the long non-coding RNAs might not be captured in the poly-A-tailed assays. What's your opinion of working with long non-coding and on single cell level, and what do you think do you miss when working with those data sets?
Yeah, well, always working with non-coding RNAs is a challenge. Yeah. Because they can be polyadenylated, they cannot be polyadenylated. They possess small levels of expression. It's always a challenge, but we do believe that the ones that we can identify here, they are actually key long non-coding RNAs, at least for the different time points of the development. And when we are dealing with single cell, of course, we are at the beginning of this new trending technology. We are only able to identify the key specific ones that possess a higher expression level. We're missing a lot, but I believe this is a nice start, a very nice beginning, that with the time, as the technology evolved, we will be able to explore deeper.
Do you have a preferred single cell technique that gives you better coverage or worse coverage of long non-coding RNAs?
No, by now we didn't try anyone. We're just working with public data. Now we are trying to acquire a 10x platform for our center. I'm actually every time here updating the grant web page, waiting for the results. And this will be a challenge actually to start to perform benchmarking analysis to see the best pipelines, actual experimental pipelines. I believe that this is the main challenge, but we are quite excited to work on it.
Yeah, data looks great. In terms of function, it's for protein coding genes, it's easier to assign a function and then maybe an action within the cell. For long non-coding RNAs, you've shown us the indirect way of co-correlation networks. What's your favorite approach and your favorite strategy there?
Oh, if we have only one candidate, we can go to the wet lab and you can play. But if you are dealing with several, with a sea of transcripts, my favorite approach is to integrate a lot of data, co-expression data, but not only. For example, the paper I presented at the introduction, we integrated several huge amount of data, similar to what the Elder presented before. I believe this is a huge power of the bioinformatics and public database, and the importance of sharing well documented data. All the metadata, they are key for that, because we can integrate the different experiments that were performed and try to add additional, to obtain additional insights. I don't know how this transcript is regulated. I don't know, you can integrate some epitranscriptome sequencing data in order to identify the post-transcriptional modifications that you can have there. You can start to assign different, obtain clues on their function. But at the very beginning, I like to start with co-expression analysis and then integrate all types of data.
There's a question in the chat about trajectory analysis and long coding RNAs. So, because that's possible, or its data is just too sparse to do that?
This is something that we wanted to work on with. We would like to work with a bigger number of cells. This work, when we started from scratch. When I'm talking about our knowledge on data analysis on single cell, because our team were working before with the bulk RNA-seq and other types of characterization. And when we started this project, if I'm not wrong, the only data set for hard development was this one with 1,300 individual cells. I believe this is a small number in order to try to obtain trajectory analysis when we are taking into account the small levels of expression of these transcripts. But this is something that we need to evaluate more deeper.
Okay, cool. So I think you also summarized your future plans.
Yes, it's another line for the future plans.
Okay, thanks so much for the great talk. Hope to speak to you soon.